Calcium and phosphate are absorbed in the GI tract. Absorption of calcium and phosphate is stimulated by the active form of vitamin D. It is a free calcium, but not phosphate. It's regulated precisely. Hormonal control of calcium levels is through the dual hormonal system, parathyroid hormone and vitamin D. Today, we will cover 3 topics. First, we will look at parathyroid hormone. Parathyroid hormone is secreted by the parathyroid glands. These are thyroid glands, and these are parathyroid glands. Most people have 4 of them. Within parathyroid glands, there are 2 populations of cells, chief cells and Oxyphil cells. Chief cells are more numerous and have a round nucleus surrounded by a small amount of cytoplasm, as shown here. Oxyphil cells are scattered among chief cells. They have a slightly smaller nucleus and eosinophillic cytoplasm. Chief cells produce parathyroid hormone, a small peptide hormone. Parathyroid hormone, or PTH, binds and activates a receptor, PTH-1R. It is a member of secreting family of G-protein coupled receptors. Activation of receptor will activate GFS and adenylate cyclase pathway. PTH-1R is expressed at high levels in the bone and kidney. 3 principal hormones control calcium levels inside the body. They are parathyroid hormone, vitamin D, and calcitonin. These hormones regulate calcium levels through 3 organs. They are the bone for calcium resorption, the kidney for calcium excretion and reabsorption, the intestine for calcium absorption. 99% of calcium is found in the bone. Most is found in the hydroxyapatite crystals. 80% of bone mass consists of cortical bones. These are cortical bones. 20% of bone mass consists of trabecular bone, and as shown here in the long bone and in the vertebrae, these are trabecular bones. Because trabecular bone has 5 times greater surface area, trabecular bone is more important for calcium turnover. There are 3 types of bone cells. Osteoblasts are the differentiated bone-forming cells, and they secrete collagen. Osteocytes are the mature bone cells enclosed in bone matrix. They are derived from osteoblasts. Osteoclasts are large multinucleated cells derived from monocytes. Their main function is to reabsorb bone. Inorganic part of the bone is composed of hydroxyapatite, while organic part of the bone is composed primarily of collagen. Active osteoblasts synthesize and extrude collagen. Collagen fibrils form arrays of organic matrix, the osteoid. Calcium phosphate is deposited in osteoid and becomes mineralized. Mineralization requires adequate calcium and phosphate, and therefore it depends on vitamin D. Alkaline phosphatase and osteocalcin play a role in bone formation. Their plasma levels are indicators of osteoblast activity. Within each bone unit are minute fluid-containing channels called the canaliculi. Interior osteocytes remain connected to surface osteoblasts through syncytial cell processes. This process permits transfer of calcium from interior surface area to extracellular fluid. Resting osteoblasts express PTH receptor PTH1R and produce RANKL. Osteoclasts express RANK, the receptor for RANKL. Paracrine signals activate osteoclasts. Osteoclasts resorb an area of mineralized bone. Local macrophages clean up debris. The process reverses when osteoblasts are recruited to the site and generate a new matrix, which is mineralized. New bone replaces previously resorbed bone. The main function of parathyroid hormone is to increase blood calcium concentration when it gets too low. It is accomplished through three mechanisms. It stimulates osteoclasts to release more calcium from the bone. It decreases secretion of calcium by the kidney and activates vitamin D, which stimulates uptake of calcium from the intestine. Osteoblasts express PTH receptor PTH1R. Activation of the receptor increases transcription of RANKL gene and repression of osteoprotein gene. RANKL activates RANK in osteoclasts. RANK is a member of TNF receptor subfamily. The overall effect of PTH is to increase plasma calcium levels and decrease plasma phosphate level. PTH acts directly on the bones to stimulate calcium resorption and the kidney to stimulate calcium reabsorption in the distal tubula and to inhibit reabsorption of phosphate. PTH also acts indirectly on the intestine by stimulating vitamin D synthesis in the kidney. The actions of PTH are summarized in this slide. When the plasma calcium level is low, PTH secretion increases. Plasma PTH levels rise. Rising PTH has three effects. It stimulates bone resorption, renal tubular reabsorption of calcium. It also stimulates formation of 1,25-dihydroxy vitamin D by the kidney. Plasma concentration of active vitamin D increases. This leads to an increase in intestinal absorption of calcium. These three effects restore plasma calcium concentration back to normal. In addition, PTH decreases renal tubular reabsorption of phosphate. This leads to an increase in urinary excretion of phosphate. As a result, plasma phosphate concentration drops. Synthesis of PTH takes place in the chief cells in the parathyroid glands. PTH is translated as a pre-prohormone. Cleavage of leader and pro sequences yields a biological active peptide of 84 amino acids. PTH synthesis and secretion is controlled by three factors. High levels of calcium inhibit PTH secretion. High levels of vitamin D inhibit PTH transcription. High levels of plasma phosphate stimulate PTH secretion. The main regulator of PTH secretion is plasma calcium concentration. This chart shows PTH secretion at various plasma calcium concentrations. Maximum PTH secretion occurs when plasma calcium concentration is below 3.5 mg per deciliter. PTH secretion is maximally inhibited when plasma calcium concentration is about 5.5 mg per deciliter. Key calcium sensor is calcium sensing receptor, CASR. A unique calcium sensing receptor on the parathyroid cell membrane senses changes in the extracellular fluid calcium concentration. The response by PTH secretion is very rapid. Calcium sensing receptor is a typical G-protein coupled receptor that activates phospholipase C and inhibits adenylate cyclase through GFI. The result is an increase in intracellular calcium levels and decrease in cyclic AMP levels. These together prevent exocytosis of PTH from secretory granules. Here is a clinical correlation. Hypocalcemia is a condition with a low blood calcium level. This condition interferes with normal muscle contraction and nerve conduction. Symptoms include paresthesia, muscle cramps, and severe spasms, along with subjective symptoms. Based on the different causes, it can be divided into 3 categories. The first condition is hypocalcemia of hypoparathyroidism. It is caused by decreased function of parathyroid glands. It could be caused by reduced or absent synthesis of PTH due to surgery or autoimmune attack. This is called the PTH-deficient hypoparathyroidism. It could also be caused by synthesis of biologically inactive PTH. This is called PTH-ineffective hypoparathyroidism. Second condition is hypocalcemia of pseudo-hypoparathyroidism. It is PTH-resistant hypoparathyroidism. It is caused by mutations in GFS. Since GFS also functions in signaling mediated by receptors for TSH, glucagon, and gonadotropins, patients are also resistant to these hormones. The third group includes conditions that contribute to hypocalcemia, such as alkalosis, congenital absence of parathyroid, as seen in DiGeorge syndrome, renal failure, and failure of vitamin D system. Hypocalcemia is a condition with high levels of calcium in the blood. It is classically summarized by stones, bonds, abdominal groans, thrombs, and psychiatric overtones. Stones refers to kidney stones. Bonds refers to bone-related complications such as osteoporosis, osteomalacia, and arthritis. Abdominal groans refers to gastrointestinal symptoms of constipation, indigestion, nausea, and vomiting. Thrombs refers to polyuria and constipation. Psychiatric overtones refers to effects on the central nervous system, such as lethargy, fatigue, depression, and memory loss. Based on the different causes, hypercalcemia can be divided into 5 groups. The first group is hypercalcemia of primary hyperparathyroidism. It is caused by excess PTH secreted from adenomatous or hyperplastic parathyroid tissue. Second group is hypercalcemia of malignancy. It is caused by excessive bone resorption by one of the 3 mechanisms. First, lymphomas. They secrete 1,25-dihydroxy vitamin D, which enhance bone resorption and intestinal calcium absorption. Second, local osteolytic hypercalcemia results from locally acting osteolytic factors released from osteolytic metastasis. Local osteolytic hypercalcemia is frequently caused by multiple myeloma in lung and breast cancer. Third is humoral hypercalcemia of malignancy. It is caused by overexpression of PTH-related protein. These proteins have marked structural homology with PTH. PTH-related protein and PTH bind to the same receptor. PTH-related proteins reproduce full spectrum of PTH activities. Third condition is Jensen's metaphyseal chondrodysplasia. It is a rare disease characterized by hypercalcemia, hypophosphatemia, short-limb dwarfism. It is caused by activating mutations of PTH receptor. Fourth condition is familial hypocalciuric hypercalcemia. In this condition, inhibition of parathyroid hormone release by calcium does not occur. It is caused by mutations in the calcium sensing receptor. Fifth condition is caused by certain medications such as thiazide diuretics. These drugs increase renal calcium reabsorption. Now let's move to part 2. We will look at relationship between vitamin D and calcium level. Vitamin D, after its conversion to 1,25-dihydroxy vitamin D3, is a principal regulator of calcium. This is vitamin D3. Vitamin D increases calcium absorption by the intestine and calcium reabsorption by the kidney. Humans acquire vitamin D from 2 sources: produced in the skin by UV radiation and obtained from the diet. Vitamin D is not a classic hormone because it is not produced and secreted by endocrine gland, nor is it a true vitamin since it can be synthesized de novo. Vitamin D is a true hormone that acts on distant target cells to evoke responses after binding to a high affinity receptor. Vitamin D travels in the blood bound to hydroxylated alpha globulin. Vitamin D receptor is a nuclear receptor. This table provides some of the genes of which transcription is either increased or decreased by vitamin D. Here, 3 genes are highlighted. Transcription of these 3 genes is increased by vitamin D. These 3 genes encode calcium binding proteins, calcium pump, and calcium channel. These 3 proteins play a central role in absorption of calcium in the intestine. Here is the synthesis of vitamin D. Vitamin D is derived from 7-dehydrocholesterol. In the skin, UV light breaks the single bond between carbon 9 and carbon 10 and convert it to pre-vitamin D3. Heat shift the double bonds and convert pre-vitamin D3 to cholecalciferol. In the liver, hydroxylation at position 25 converts cholecalciferol to calciferdiol. In the kidney, hydroxylation at position 1 converts calciferdiol to calcitriol. Calcitriol is the most active form of vitamin D. This information is summarized in this slide. Here is regulation of vitamin D synthesis. The 1-alpha hydroxylase is the major point of regulation. PTH increases transcription of 1-alpha hydroxylase gene. Phosphate inhibit 1-alpha hydroxylase enzyme. Through a negative feedback loop, vitamin D3 decreases transcription of 1-alpha hydroxylase gene. If excess vitamin D is produced, it stimulates a gene coding for 24-hydroxylase. This enzyme inactivates vitamin D by hydroxylation at position 24. Finally, let's look at calcitonin. Calcitonin is a 32 amino acid polypeptide hormone. Here is a follicle from thyroid gland, and this is a parafollicular C cell. Calcitonin is secreted from parafollicular C cells when blood calcium levels are high. Calcitonin lowers calcium levels by slowing the calcium-releasing activity of the osteoclasts in the bone and increasing calcium secretion by the kidney. Calcitonin binds calcitonin receptor, a G protein-coupled receptor, and activates both G-alpha-S and G-alpha-Q. Calcitonin acts mostly during childhood